Hey guys, welcome to the Bio 25 review. It is Friday, February 12th, and today we are going to cover content from week 2. Well, we're going to cover the other half. If you're interested in watching videos on the first half of week 2, which was about KD, and I believe like protein interactions, like environmental factors and protein interactions. It's the video before this. I believe Jason and Rachel did that review. So without further ado, me and Tina are gonna just jump into this review session. Cool. Tina, you want to do the slides and like switch off? We can do that.
Yeah, sounds good.
Yeah.
All right. So for a little review of the protein ligand interactions, ligands are essentially, it's a broad term where it is anything that a protein binds to. So in the picture on the side, in this case, the CAMP or the cyclic AMP, that's a ligand. And then it binds into the binding pocket of a protein, but it doesn't always have to be a pocket like the picture is showing. It can just be like a flat surface, something that the ligand can interact with and bind to. So that's just something to keep in mind. And then it's always important also to understand that when coming to protein ligand interactions, it's really important that the protein and the ligand are oriented correctly, so that all the amino acids are exposed the way they should be, because otherwise the ligand won't be able to quote unquote like fit in properly. And so this also comes into play the R group interactions. So the hydrophobic or van der Waals interactions, ionic bonds, dipole bonds, hydrogen bonds, ion dipoles, everything that we were talking about last week. And so this is a huge part because it comes in play where if it binds in, that chemistry will determine how well that they bind together. The size also matters. If it's too large, the ligand also won't be able to bind as well. And then, kind of the idea of structure and function, where the structure of the protein and then the structure of the ligand, of course, that leads to determining what the protein ligand and complex does as its function. And then the next slide.
Cool.
Okay. So for the affinity, we talked about this in class today too, I think, but affinity is the strength of the interaction between the protein and the molecule that it's binding to, or the ligand. It depends on two main things. One, the number of bonds between proteins and the ligand, and then two, the strength of these bonds. Usually the bonds are non-covalent bonds, just because it allows the cell to be more dynamic since the covalent bonds are stronger. And then the type and angle of the bonds for greater affinity, it all depends on the type of R groups that are there. So again, bringing back the chemistry of the amino acids within the protein and the ligand, that all incorporates into how good of an affinity or how bad of an affinity the protein and the ligand have together. But keep in mind, of course, that just because the affinity is strong or weak, it doesn't correlate directly to if that's a good or a bad thing. So the stronger the affinity, that doesn't necessarily mean it's a good thing. It's all dependent on what the cell needs and what the function of that protein interaction is needed in the cell. And that was called the Goldilocks hypothesis.
Excuse me, can you say the good, like two sentences back again about the Goldilocks hypothesis?
Yeah, so I don't know if you know the story of Goldilocks, but basically it's the story of the girl who has like 3 bowls of oatmeal or something, or porridge, but it's the idea of you don't want something that's too good and you don't want something that's too bad. It's all about what you personally need and what this, in this case, what the cell needs, so that's just right. So just because Just because the affinity is stronger or weaker doesn't mean that it's what the cell needs. It's all about, it's very unique and particular to each cell.
Oh, okay, thank you.
No problem.
Yeah, Tina, I was gonna say, do you want to do like all the protein ligand stuff, and then I can just do like the next section rather than switch off?
Oh, yeah, I'm sorry. I thought that's what we were
I was
doing.
just wanted to make sure, but you're good. You're good.
Sounds good.
Perfect. That was perfect.
All right, and then relating to the affinity, we have the way that we measure the protein ligand interactions, and then their affinity is by the dissociation constant, which is labeled as KD. And so the dissociation constant is an average rate measurement of how often the proteins dissociate from its ligand. So how often the protein and the ligand are not connected to each other or bonded to each other. The equation for KD is in this top corner here, where it's P times L divided by C, where P is the concentration of proteins, L is the concentration of ligands, and then C is the concentration of the complex. And then complex just means when the protein and the ligand are bonded together. And then essentially what this KD is looking for is the concentration of ligand when half of the binding sites of the proteins are filled. So when you have half complex and half free protein, that's also P is equal to C. But the simplest way to think about this, in my opinion, is that the dissociation constant and the affinity are inversely related. So the smaller your dissociation constant, the tighter the protein and ligand interaction. So the higher the affinity. And if you think about it, it makes sense, because if your dissociation constant is really small and really low, that means it's not likely for the protein and the ligand to separate. So then that means that they fit really well together. So their affinity is really high. Next slide. And then just another little analogy to help you guys better understand. The dissociation constant, enzymes are a really good way of thinking about it, where essentially, since enzymes help a lot of reactions go along, you want the enzyme to be able to bind strongly to a substrate or the reactants. And so you would want the KD to be lower in this case, because in order for the enzyme to be securely attached, it has to have a high affinity. So then high affinity, you have low KD. And then on the flip side, once the product is formed, the enzyme, you no longer want the enzyme to be able to have a high affinity, because otherwise the products won't be able to leave the enzyme and then go do what it needs to do, and the enzyme won't be able to be recycled either. So once the product is formed, the KD is now higher, and then if your KD is higher, that means it's much more likely for the enzyme, it's much more likely for the enzyme and the ligands to dissociate. Oh, sorry. And then this is just a visual of the enzyme analogy. So that first part, that you have the enzyme and then the substrate is coming in. So you would want a high affinity in this case. So then that would be a low KD so that they bind together. And then once the enzyme goes through catalysis, does what it needs to do, it forms this product, which is shown by the blue circle. This actually changes the chemistry, which is how the KD and then the affinity changes. But once this product is formed, you want it to be released. So now the KD is higher, so they dissociate, and then the affinity is lower.
Okay. Thank you, Tina. Now I'm going to jump into the next category for this review, which is enzymes. So what are enzymes? So if most of you guys probably have taken general chemistry, Gen Chem, or are taking Orgo right now. But enzymes are essentially catalysts that carry out chemical reactions. And enzymes are involved in these reactions by making or breaking bonds. And they're able to form and destroy covalent bonds. And so this was just a little throwback to when we learned on week 1 about peptide bond formation from enzymes. Enzymes are able to break these types of bonds because they're covalent. I'm trying to think if enzymes also break non-covalent bonds. Tina, do you remember?
Sorry, what did you say? If enzymes can also?
Break non-covalent bonds. Just wondering if I
Um,
made a typo in here.
I know that they mostly do covalent bonds, but I'm not sure if they do non-covalent bonds since.
Yeah, but I'll look into it and get back to you guys on that. So sorry. But yeah, like I was saying, enzymes are involved in the formation and destruction of covalent bonds. And examples of covalent bonds we've seen in this class are peptide bonds in your primary structure and disulfide bridges, which are involved in tertiary quaternary structure. Ribosomes form covalent bond peptide bonds between amino acids, and spliceosomes break covalent bonds between sugar molecules. Enzymes, why are enzymes important? Why do we need them? Enzymes are vital to cells because they change the chemical structure of molecules. And enzymes themselves are actually proteins, so enzymes can be regulated. And I think we'll get to like regulation of enzymes in the upcoming slides. And it's important to know, a lot of people operate under the assumption that every single enzyme that you see in science is 100% protein, but however, not all enzymes are made this way. There are some enzymes that are actually made of like a mixture of RNA and protein. And the RNA serves as the catalytic molecule that's involved in the catalysis or the enzymatic reaction. And those are called ribozymes, or RNA enzymes. And yeah, you can see, let me see if I have the pointer. In the bottom diagram, enzymes either form or break covalent bonds. So you can see how you have the substrate going into the active site. It forces them and forces their bonds and causes it to reach a transition state. And then your product is formed, and then your products dissociate from the enzyme. So in general, the KD of the substrate of the enzyme substrate complex, I believe, would be lower because they have high affinity compared to the KD of the products, because their products, you want them to dissociate very quickly from the enzyme so that that enzyme can be reused again and repurposed for a different reaction. So yeah, that's what enzymes are. Does anyone have questions so far? Or Tina, do you want to add anything?
Oh, I was trying to find if enzymes do break non-covalent bonds, and I couldn't find it either. So maybe we could ask that later on.
Yeah, we can ask later on, but I'm pretty
Yeah.
sure it's covalent. I don't know why I was second guessing myself. But yeah, so this is just a slide that explains what are enzymes. And I actually have a video clip for you guys. I watched this video for, I think, Biochem 420. And I think it does a good job of explaining enzymes and like what goes on in the active site, but you're not responsible for like the super molecular details it gets into at the end, just sort of having a general idea of what enzymes are. So the content from your notes will help you the most, and like just general content from the slides too. But this video, if it gets like too detailed in this stuff, just don't panic. Yeah.
The lysozyme enzyme cleaves polysaccharide chains. First, the enzyme and substrate associate, forming an enzyme substrate complex. The enzyme catalyzes a hydrolysis reaction that cleaves the substrate into products, which are quickly released, allowing the enzyme to catalyze another reaction. The cleft in the enzyme holds 6 sugar residues of a polysaccharide. The hydrolysis reaction occurs between residues. Looking at the details of the reaction in solution, the sugar residues adopt their most stable 3-dimensional conformation. However, after the polysaccharide enters into the enzyme substrate complex, the enzyme forces the sugar shown on the left into a strained conformation that more closely resembles the transition state of the reaction. 2 amino acids within the enzyme facilitate the reaction. A glutamic acid donates a proton to the sugar on the right, and an aspartic acid attacks the C1 carbon atom of the sugar on the left. The attack on the C1 carbon results in a transient covalent bond between the sugar and the amino acid, and hydrolysis of the sugar-sugar bond. The deprotonated glutamic acid then polarizes a water molecule, drawing a proton away from it. This allows the water oxygen to attack the C1 carbon, breaking the sugar aspartate bond. In this way, the 2 amino acids are returned to their original states, forming the enzyme product complex. The enzyme and products dissociate.
Yeah, so as you can see, that's just an example of enzymes in the real life example, the lysozyme enzyme, but the specific molecular details you don't need to know, but I just wanted to give you guys an example of what it looks like in real life. And you can see from the video, I have the enzyme substrate complex and how it has to be in close contact with each other to be able to, for the enzyme to be able to perform a reaction on the active site. And then once you have the products, they form and dissociate. And yeah, it's just the video. Don't know why it won't let me exit out of it. Sorry. Does anyone have any questions?
No.
Okay. So just going to continue talking about enzymes. So just like I said, enzymes bind to a substrate for catalysis or the enzymatic reaction to occur. So to be able to do this, they have to have some affinity for the substrate. And during reactions, enzymes have the ability to undergo conformational shifts, but they ultimately remain unchanged at the end of the reaction. So the reason why they undergo conformational shifts is so that they can accommodate the substrate, which is in a specific shape. So you can see that the active site sort of has the same hexagonal shape that the substrate has, so it's able to fit it in. And this is just like reviewing affinity and why we would expect enzymes to have high affinity for the substrate. Or substrate is also another word for reactant. So ideally we'd want our enzyme to have high affinity for the reactant because it will latch onto the substrate, and then when it's done catalyzing the reaction, and meaning that the enzyme turns the substrate into products, then at that point, once you have the enzyme product complex that is formed, you want the enzyme to completely dissociate itself or completely separate itself away from the product, because you don't want the enzyme and product to be stuck together since the product has been made and it needs to go elsewhere into the cell. So the enzyme at the end of the reaction will release the product, and then enzymes are reusable. So what will happen is it will release the product and then it will find some new substrates or new reactants to redo the reaction over and over again. And yeah, so that's why enzymes have high affinity, or if you think about it in the inverse way, they have a low KD or a low dissociation constant. So does anyone have any questions about that? No? Okay. Why are enzymes useful? Enzymes are useful because they increase the frequency of chemical reactions, and they ensure that they happen faster. They happen faster than the reactions, the reactions happen faster than they would if they were to occur spontaneously or without an enzyme. And another reason that enzymes are useful is, I don't know, I think it's pretty much the same thing 1 and 2, but just like I said, the time for a spontaneous reaction without an enzyme, so for the cell to just perform reactions on its own, takes too slow. And enzymes are useful because they can be regulated. So cellular processes are regulated. And when cell processes are regulated, this ensures that they're performed correctly and the cell is able to function properly. And so here I just have a diagram of just a basic enzyme reaction scheme. And it shows the time on the x-axis and free energy on the y-axis. And it just shows the reaction progress over time. And yeah, so that's just the graph. And you can see that in an enzyme is a catalyst. So you can see that without a catalyst, it takes a lot longer for the reaction to reach the transition state. Whereas with the catalyst, with the enzyme, it doesn't take as long to reach this point, which is the transition state. And so I tend to think of an analogy kind of like when you're biking up a hill. It's a lot easier to bike up this hill because it's smaller, takes like less, it takes like less effort on your part to bike up the hill, and it's a lot easier to get to the top and back down again. So that's sort of similar for enzymes. So with the catalyst, it's easier for the reaction to occur and makes it more frequent that reactions can occur because the transition state's not so high, whereas without an enzyme, or if you were biking up this hill, it's a lot steeper and it will take you a little bit longer to get up to the top. And so that's why those reactions don't recur as frequently without a catalyst. And yeah, does anyone have any questions? Okay, I'll continue. So just like I said in the other diagram, there is a transition state that the reactions must go through to turn into the product of the reaction. And a transition state is just the top of the hill. So this area, in this area. And transition state is highly unstable and it's a high energy state. And the activation energy is the difference between the transition state and the energy of the reactants. And so it actually shows in this diagram that the energy of the reactant and the transition state, like the difference between those two, is called the activation energy. And so essentially what enzymes do is they lower the activation energy, and that enables the reaction to occur much more easily. And so you can see like with the catalyst, the hill is a lot smaller because the activation energy is lowered. And yeah, so sorry, I keep flip-flopping between the slides, but I just wanted to point that out. So enzymes, they lower the activation energy of a chemical reaction and they help to stabilize the transition state. And stabilizing the transition states what speeds up the reaction. And this is because enzymes ideally want the reactions to reach the transition state. And when the free energy of the reactants is larger than the products, then the reaction is said to be spontaneous or energetically favorable. And the reaction is also considered exergonic because energy is given off in the reaction or energy is released. And inversely, if the free energy of the reactants is smaller than the products, then the reaction is considered to be not spontaneous or non-spontaneous. And we would call this an endergonic reaction because energy input is required to create the products. Okay, anyone have questions? Okay. So then this is just a slide that sort of summarizes what I said before. It's beneficial for enzymes to lower the activation energy because the reaction is able to happen a lot smoother and faster. And if reactions happen quicker, then it's like more like there's a fast reaction time and it's more energy efficient. And you can see just like the other diagram that because with an enzyme, you have the lowering of that lowers the activation energy. So the difference between the reactants free energy and the transition state. So it makes it a lot lower. So then the reaction is able to proceed more smoothly. And it's a lot faster for it to get up to the transition state, which means this is the point where the substrate turns into products. And yeah, that's just enzymes. More about enzymes, how exactly they work. Laura goes over, I think, 3 main methods about enzymes. One of them is that the enzyme will bind to substrate molecules and orient them precisely enough to encourage a reaction to occur between them. Second method is that the binding of substrates to enzymes causes a rearrangement in the electrons within that particular substrate, which creates partial charges. And these partial charges favor reaction. And then the third way that enzymes can work is that enzymes have the ability to strain the substrate molecules or the reactants, which force it towards a more favorable condition or transition state. And that helps to make the reaction more favorable. And yeah, does anyone have questions so far? Okay. Tina, you want to do this next part? Tina?
Oh, sorry. The regulation of protein. So clearly before, there's a lot of information on enzymes, and I know that some students, they reached out to me saying there was like, they felt a lot of content was thrown out, but hopefully all that information on enzymes is helpful because they are really important. And then there are different ways that the proteins are regulated. So one way would be allostery. It's not a term that Professor Francis uses very often. However, it is good to know it still. So again, it's regulatory, and what it does is allows for a molecule to bind to a site of a protein other than the actual active site. And then that causes the enzyme to change shape or a conformational change that will then affect the enzyme's ability to perform its job. So as you can see in that picture on the side, the allosteric site would be that second binding site where the inhibitor, or in this case, the inhibitor, but any other ligand can go to bind to, and then that'll change the shape of the enzyme, which then causes the function to change. So the main idea of structure equals function again. So in this case, if that allosteric site was not bound to, this green substrate would have been able to fit into that active site. But since the allosteric site was bound, the active site is now altered, and then that substrate can no longer bind in. So it's almost like it's almost like regulating from a distance, and more or less. And there are different binding sites and types of allostery, but Professor Francis does not really go into the specifics, so you guys don't need to know that either. And then the next one. Another way for regulating is post-translational modifications. So these happen after the protein is made, hence post-translational. So after the tertiary structure is made and everything. In this case, functional groups are added covalently. Popular ones would be phosphorylation, ubiquitination, or acetylation. And then these are also regulated by an enzyme, and they are reversible. So an enzyme can remove these as well. So an example would be like kinase is an enzyme that can phosphorylate substrates. And then to reverse it, you can use like phosphatase to dephosphorylate something. And essentially, it's a switch mechanism where you change the protein surface by adding these functional groups, and then by changing the surface chemistry, you thus change its structure, which again changes its function, and then essentially you change what the protein can or can't do. And then again, it is reversible by removing that modification. And then on the side, it just shows you a picture. The yellow is phosphorylation, green is ubiquitination, and then the little triangle blue ones are acetylation. So you just add them to these proteins after they've been translated already, and then thus the surface chemistry changes, and then the way that the protein as a whole that the chemistry changes, which then changes the function. And then the last type of protein function regulation that we talked about is nucleotide binding and hydrolysis. So these can lead to different binding and active states. They are caused, or they can be done through GTPases or ATPases. Those are just categories of enzymes. So GTPases bind with GTP, and then ATPases have to do with ATP. They can't like interchange or anything. That's something to always keep in mind. And so it's the ability to change or cleave bonds between the phosphates and then change GTP to GDP. And then that in itself is a conformational change, which leads to a change in activity, because when you change the shape, again, that changes the function. And then it's also a regulatory mechanism that's really common in a lot of different pathways. This will definitely become more relevant and more important as we go on later in the course. I think in the next couple of weeks, maybe, I'm not positive, but I think it's soon. But essentially, GTP and GDP, or ATP and ADP, depending on what you're looking at, they are these switches that when they bind to the proteins, they either activate or deactivate them. In this example, you can see that the green is a binding protein, in which when the GTP is on that protein, is bound to the protein, it is active, it's on. And then it goes through hydrolysis, in which the GTP dissociates, and then a GDP binds in, and then that flips the switch, causing the protein to be now inactive. And then just to finish that picture, the GDP can also disassociate out and diffuse out, allowing the GTP to go back in, and then it'll be active again, so on and so forth.
That's pretty much it. That's the review for today. Please feel free to reach out to us if you have questions. And that's all the material we have for you today.
Thank you.